Good morning. My name is Jake, and today I'll be presenting my fifth year undergraduate thesis, which I completed in the Juliet Daniel Lab in the Center for Discovery and Cancer Research at McMaster University. It is entitled The Complex Story of Kaiso Lessons from Genetically Modified Tumor Cells. Every 2 in 5 Canadians will be diagnosed with cancer at some point in their life, and almost 100% of Canadians will be affected by cancer in some way, shape, or form. Colon cancer is a special subset of cancer that affects the intestines and the colon, and it has a very high prevalence and death rate in Canada. Typically, we define cancer as being an uncontrollable growth of cells, which are the building blocks of our body. Within each of these cells exists something called DNA, which is the genetic blueprint that tells our cells what to do and where to go. But sometimes when the body makes a mistake or this pattern is interrupted, what happens is we have a mutation. Mutated DNA can no longer tell our cells how to behave appropriately, and as a result, we see the development of cancer. My lab discovered a very important protein known as Kaiso that is suspected and has been shown to play a very big role in the development of multiple cancers, including breast, lung, prostatic, and colon cancer. When we see lots of Kaiso, we typically see lots of cancer. So if you have a lot of Kaiso in your body, you may be predisposed to developing cancer at some point in your life. However, something very interesting happens when you take these tumor cells outside of the body. In these isolated cancer cells, when you increase Kaiso, it kills the cell. But how does that make any sense? If Kaiso promotes cancer, why does it kill cancer cells when it's increased? Well, a second important protein known as p53 was shown to collaborate with Kaiso to kill cells in the isolated environment of a petri dish. But when those cells are inside of a living animal or a human, the environment around the cells can supplement them and keep them alive. This poses a very interesting problem for researchers. Since it's unethical to use hundreds of human or animal subjects, we rely on petri dish studies, commonly referred to as in vitro studies. My main thesis aim was therefore to genetically modify a special type of colon cancer cell that lack the p53 protein that collaborates with Kaiso to kill its cells. Then I wanted to permanently increase the expression of Kaiso to watch how it affects cells long term without killing them. In other words, I needed to figure out a way to successfully increase the amount of Kaiso in these cells without them dying. To do this, I used a very special tool known as CRISPR to artificially increase the amount of Kaiser present in these cells with or without the p53 protein. In short, CRISPR uses the immune system of bacteria to turn on, turn off, or delete a specific piece of DNA that we tell it to. By turning on the Kaiser DNA, I was able to successfully increase the expression of Kaiser in these cells. Currently, I'm still performing the experiments that will show if my special cells are protected from too much Kaiser. If my hypothesis is correct, that means that our lab will be the first to create a stable cell line that does not die when Kaiso is increased. This will provide researchers with a more informative model of Kaiso in cancer, and will offer critical insights into Kaiser's role as a predictive marker or a drug target for cancer treatments moving forward. Given the rise of rates of cancer in the Canadian population, it is important for studies like this to be performed to give Canadians the best possible outlook on their health moving forward. Thank you very much.